Hi, this is Miss Litton, and this is the final official 7th period review that we are having this school year. So this is chapter 24. It is fairly brief because this is the half of the chapter where we just discussed biotechnology and genetic engineering. And then that's it for this particular unit because most of the days were the Amgen lab. And I will tell you here and now that on the final, I asked about biotechnology that's from the content. I did not ask an Amgen specific question. Okay, but I asked you about biotechnology. So once we finish this review, if you want to hit chapter 27 of all the chapters on your final, that one has the most questions, is chapter 27. So I'm happy to go over any big ticket items there. If you need more time with chapter 27, what could you do? Yeah, watch the chapter specific review. So the first thing I want you to try to do when you think about biotechnology is we learned about words like cloning and plasmid, and we learned about some enzymes. Whatever you're doing your review on right now, see how many of those big ticket words you can come up with, the different types of enzymes and what they do. See what you can pull out of your head right now. And I'll pause this just for a moment and get you some licorice. Okay, we have our licorice now. All right. So the first thing I want to talk to you about would be cloning. When you have identical copies of something, you could have identical copies of an organism, or you could clone a gene and have identical copies of a gene. When you work with recombinant DNA, what do you hear in recombinant? Recombine. So we have new combinations. And you have to have a vector of some sort and something that the DNA code, what you're going to be using. And usually it's a plasmid. Could be also a virus, which is an extra ring of DNA. And remember, DNA is a code book. So as long as you have the code book, you can do, how do you get a protein from that code book? What's step one?
Heat it up.
Starts with a T. Sounds like yesterday at lunch. Transcription. When you go from DNA to what? RNA. And then when you use that RNA to build a protein, that's called translation. Okay, yay. All right. So then we looked at how you go about doing that. Welcome, we're eating licorice. So here's a human chromosome, and you might want to cut a gene out of it. What are the chemical scissors that we use?
Restriction enzymes.
And restriction enzymes cut at a specific series of bases called a restriction site. Another name? Recognition sequence. Oh, I recognize you. I'm an enzyme. I recognize you. I will cut you. Okay, so you can cut that out. And as long as you cut the plasmid and the restriction enzyme, the plasmid and the two pieces of DNA with the same restriction enzyme, then the same sticky ends will be there. Do you remember what sticky ends are?
They're like matching.
Complementary.
Yeah, here I'll show you a piece of sticky ends. Right here. So when the restriction enzymes, this wouldn't work if you had a blunt cut. This only works if you're coming across the DNA and you're exposing these base pairs because they're going to want to make a hydrogen bond with a complementary base, right? A with T and G with C, right? So these are sticky ends. So since restriction enzymes cut at a specific series of bases, they will have these sticky ends. If the sticky ends are the same, then you can mix and match DNA. Okay, so that is one key thing you have to have for genetic engineering is you have to have restriction enzymes. Then when you put them back together, yes, the hydrogen bonds will hold the bases together, but you need to seal the phosphate sugar backbone. And what enzyme do you use for that? Ligase, right? Remember that part? Okay. And that way you can mix and match DNA from different sources. All right. Then you can give that plasmid, that genetically engineered plasmid, force it into a bacterial cell. What is that process called? It starts with a T when you force DNA into a cell. Transformation. Boom, shakalaka.
Watch out.
Save some for us.
Yeah.
All right, transformation. So you transform. Remember you had a lab on that?
Yeah.
Okay. Yeah. All right, it was lab 5.
Yeah.
Okay. So you're transforming the bacteria with this plasmid. Now it will transcribe and translate that DNA. These are clones of this original one, right? And they're all making that product which you can then harvest from that bacteria. Okay, I'm moving on. So then the idea was, what if you only have small amounts of DNA? Do you remember what you do to make more DNA?
Replicate them.
What?
Do you like replicate them?
Yeah, how? Man-made. How does man do that? Well, DNA polymerase is the enzyme, but how can we, if I want more copies of a DNA, like say for a crime scene, and I need lots of copies, there's a machine I use and there's a process.
The printer thingy.
Yes, printer thingy. PCR. Yeah. Polymerase chain reaction, and it's done in a thermocycler. Okay. And in order to get that to happen, you have to heat up the DNA because you don't have any what? Helicase, yeah. Because if you left helicase in there that was active, it would just constantly unwrap the DNA. So you can't do that. So you use heat to unwrap the DNA. That's helicase. Then you use an enzyme. Typically, they use something from an archaea, Thermus aquaticus, because it can withstand high heat temperatures and not get denatured, and you just replicate it. And you go through these heat and cool cycles until you have millions of copies of the DNA. Okay, so that's called a PCR machine. Now I have millions of copies, and I cut up the DNA. Maybe I'm looking at STIRs. Remember STIRs? Yes, you do. Tandem repeats. Do you remember that? Okay. So let's say I cut up the DNA. What's a process where I can see how big the fragments of DNA that...
Electrophoresis.
Yes. Okay? And so you load it into a gel, you apply electricity, DNA is charged negatively and it's attracted to the... Yes, and which pieces move faster?
Smaller.
Mm-hmm. Perfect. And then you can find a banding pattern, and the banding pattern for your DNA is unique to you unless you are a identical twin. Okay? Good. We look like this, and this is a sad little crime scene, because really, why do they have that? But so they've cut up the DNA, put it in a gel, and then run the gel. Okay, so that would be DNA profiling. Then some other ways that DNA is used in biotechnology. So you're using in biotech, you're using the processes, the metabolic processes of a cell or an organism in order to get something that you want from it. So we talked about genetically engineering cats, right? So that people wouldn't be allergic to those cats. Genetically engineering bacteria, so it'll break down oil in case there's an oil spill, or it will mine minerals out. It maybe will make plants, if you sprayed the bacteria on the plant, maybe you could make it so that pests wouldn't grow on the plant. Lots of different options there. There's the biodegrading. You could genetically engineer the plants themselves if they generated their own insecticide, or you make them frost resistant or drought resistant, or you can have them water them and maybe with a higher amount of salt water. Here's a genetically engineered tomato. So it's a tomato on top and a potato in the roots. And maybe we could genetically engineer plants to carry medicines for us, so that you could grow those medicines up if you didn't have access to them, things like clotting factors. Then maybe we could make some transgenic animals that made products for us that we need. Maybe growth factors or things, and we can get animals to generate them for us. Okay, so we didn't do all of chapter 24. Part of it we did before. We did the parts of 24 from here on. And could you address all of those different topics from here on? Yeah? Okay. Now, any questions on chapter 24? Would you like me to review chapter 27?
Yes, please.
Sure. Okay, let's do it. This is how we do it now.
test, are they gonna be like the math problems?
Yes, Hardy Weinberg will be on there. Be Hardy ready. Angus, you don't get any more.
Oh my, I had like 6.
Oh, we're started already. Sorry, I forgot. Ready?
Yeah, ready.
Anything I ask you on plate tectonics would be really easy. So don't stress about that. Okay? Plates move, right? Why do the plates move? Convection currents underneath, right? When two plates meet, that would be a what kind of boundary? Convergent, right? And if it's two continental plates, it would go up and form a mountain, right? And otherwise you would have a what kind of zone? And then you could get what right there? Volcanoes. Good. All right. And you could have a divergent plate as well. Okay, we talk about this later when we talk about the age of fossils, but you can look at rock layers as well, and you can tell by the amount of percentage of an isotope that you have, you can tell how old it is. That would be absolute dating or carbon dating. And here again is a little bit more on plate tectonics. When we look at the continents kind of fit together like what?
Puzzles.
Puzzle pieces. So why is it, keeping that in mind, why is it that a lot of reptiles and amphibians are the same, but mammals are so different on all the continents?
Because of environmental changes make evolution.
Think about the plate tectonic theory to answer that question. When all the continents were together, that's when reptiles and amphibians and all of that were evolving. When the continents had separated, that's when mammals evolved, and that's why you have things like koala bears in Australia and giraffes in one place, horses in another place. Because when mammals were evolving, the continents were already separated from each other. Right? Okay? But as long as the continents were together, as long as land masses were connected, whatever was evolving, those could match and line up. Yeah?
Yeah.
Okay. All right. Then we talked about early Earth. Early Earth, in a nutshell, early Earth at first, the early gases were gases that came out of what? Volcanoes, right? And then there was water, but there was no. So what kind of environment was it? What do you call a non-oxygen environment? Starts with an R. Not oxidized, but reduced. It's a reducing environment, okay? No free oxygen. The gases that came out were like gases out of a volcano, and no free oxygen, which is good because it was conducive to building. And we looked at this step, like how life could have evolved. And so we said these first inorganic molecules right here would be the gases that came out of the volcano. And then here, these inorganic molecules would have to become organic molecules. What's that right there? Amino acid, sugars, fatty acids. How could we get from this step, how could we jump up to this step right here? What would we need? Energy. Where could we get that energy from? Lightning, what else? Solar energy, molten magma, radiation, right? And it was hypothesized that if you had that type of energy, you could go from inorganic molecules to organic molecules. Operin hypothesized it, but a man did an experiment to show that. What was the name of that man? No. Sometimes people drink this after a long day. Beer, name of beer. Miller, there you go, baby. All right. So Miller, okay. Miller proposed.
Do not drink.
Calling your mom right now. Miller, remember the apparatus? And he cycled the gases around, and in just 1 week's time, he got organic molecules. So we're talking millions of years here, right? Because the formation of the Earth was about 4.5 billion years, and the first fossil is about 3.5 billion years old. So you have 1 billion years to form the first living organism. 1 billion years. Okay, how many years have you lived? 16 years. In a week, he was able to do the first step. Go from inorganic molecules to organic molecules. Okay, once you have these organic molecules, you need to go up a step and you need to make macromolecules, polymers. Polymers like what? Proteins and carbohydrates and lipids and nucleic acids. And there are hypotheses about that too. That when hot aqueous solutions cool, that you can form proteins, or things called proteinoid microspheres. That clay could act as catalysts. Again, the energy we talked about earlier could form those polymers. Then once those polymers are formed, you have to build your first cell. Was your first cell, do you think, a prokaryote or a eukaryote? Yeah, it was probably a prokaryote because that's simpler, right? And then was it an autotroph or a heterotroph? Yeah, it's easier to be a heterotroph than an autotroph, right? Because heterotrophs just consume nutrients. Autotrophs have to make the nutrients and then use them, right? Okay, so the first cell, so to get from here, from these proteins and lipids to cells, well, lipids will cool and form spheres, so will proteins. So it's possible that you had this early sphere of lipids or proteins, and then the reactions that took place inside that early cell were different than the reactions taking place outside. You would need some sort of genetic code book, probably RNA, and there's more detail there than you would have to know. But the idea is that you would build these polymers and you would form cells that were heterotrophs, and then later you would have autotrophs and you would have eukaryotes, right? Remember we talked about the endosymbiotic hypothesis where one prokaryote lives inside of another prokaryote, right? Okay, so that would be steps to life. And then the next thing we talked about was evidence of evolution. Do you know what evidence is there based on those pictures? What would this evidence be right here?
Fossils.
Fossils, good. What was this one? Biochemical, comparing DNA, comparing the proteins it codes for, the sequence of amino acids in the proteins. Remember you did a lab on that, or a POGL on that, or comparing the sequence of bases in DNA. Okay, so fossils, this is the one that's used most currently. Remember we formed a cladogram, all the descendants. What was this about? Biogeographical evidence, good. What's this about? Anatomical. What were the 3 anatomical pieces of evidence? Who was here yesterday at lunch? I would ask a question. Everybody would be like,
oh yeah.
you remember homologous structures? I heard it. Did you say that? Okay, he's all, that was me. Homologous structures, that would be what you're seeing right here. The bones are the same. If the bone is the same, then probably the DNA is the same, right? Homologous structures don't have to do the same thing. That's analogous, and that is not evidence. Just because dolphins swim and fish swim, it doesn't mean that they're related. Butterflies swim for a very short time. Butterflies and birds fly, but they are not similar structures, right? So they're not probably related. So homologous structure, if you have the same structure, same kind of anatomy, you may use it differently, you're probably related. What's another piece of anatomical evidence? Vestigial structures. They point to your ancestral past. That's a piece of evidence. Remember I told you that whales have what kind of bones? Hip bones, little femurs, that you'll see salamanders in caves that have little vestigial eyes. You have a tailbone, right? Those are all vestigial structures pointing to our past. And then the last one, baby, something baby, starts with an E. Embryo, yeah. When you compare embryos, so embryonic evidence, okay? More similar the embryo, probably the more closely they're related. And also when you look at embryos develop, sometimes it's evolution on fast forward because we have what during our development? Gills and a tail. Good. And then this was the geologic time scale. Remember this one? What era are we in right now? Cenozoic. And that's now. Back how many? 65 million years. Okay? And then before the Cenozoic was Mesozoic in the middle. It ended 65 million years ago, but when did it start? Yeah, sometime after school. Just think about like that. You can say 248, 225. I'm actually good with all of those things. You'll get it right on your test. And then before the Mesozoic was Paleozoic. Think old and pale. And that's when you had a huge burst of life on the scene. A lot of things happened during the Paleozoic era. And that ended about 540 million years. And everything before that is Precambrian. And that's in the billions, right? The by the time you are at the end of the Precambrian, Precambrian, you had early invertebrates at that point. You had algae. That's where you start off. And then you enter into the Paleozoic, the old and Paleozoic era. And then you go from algae all the way to gymnosperms. And you go from invertebrates all the way to early reptiles. So that is a huge amount of life evolving then. The Mesozoic era was the time of, for animals, yeah, reptiles, good. And for plants, it was a whole bunch of gymnosperms, right? Christmas trees, Hanukkah bushes, right? Then at the end of the Mesozoic era, that's when you first got mammals, early mammals, and you first got flowering plants, right? And then your Cenozoic era is the time, remember we talked about when you look at the whole geological time scale at the very end, who evolved? Humans, yeah, last 30 seconds, right? Okay, that was all that. Again, if you want more details on this, go back to the whole review I did just on chapter 27. As far as speed of evolution, you remember the two? Gradualism, phyletic gradualism, and punctuated. Punctuated would say that things happened rapidly and then it was status quo for a while and then rapid changes. Phyletic gradualism says it was slow and steady. Which one would you have the most transitional forms? Gradualism, good. Okay, we did all of that. All right, so cladograms. So you have this ancestral species and then you see all of its descendants, yes? Okay. Okay, now I want to talk about we talked about micro and macro evolution. Which one has more which one has a new species? Macro. Okay, good. So then we talked about the 5 fingers of evolution, what causes change. Remember that? What was the pinky? Small populations. Now, when you have a small population, you're more likely to have genetic what? Drift. What were the 2 kinds of genetic drift? Bottleneck, which is I went through a crisis. What's the other one? Founders. Okay, good job. So when we talk about Hardy-Weinberg, it's saying Hardy-Weinberg is kind of set up to fail, right? Because it says you have to meet these 5 conditions. You have to have all your populations be large, because if a population is small, you will have genetic drift, right? So what Hardy-Weinberg says is if you want to keep your allele frequency exactly the same, like if big T is 0.7, then little t is going to be what? 0.3. If you want to keep your allele frequencies the same, you have to meet these 5 conditions. You have to have large populations. You have to have no selection. You have to have no mutations. You have to have no gene flow. You have to have no natural selection. If you have none of the 5 fingers, then the allele frequencies will stay the same and evolution will not occur. But if you have any of the 5 fingers, then evolution will occur. Can you ever stop the middle finger? What does the middle finger stand for? Can you ever stop mutations? No. So since mutations happen, allele frequencies will change, and therefore evolution will occur. You got that? It's setting up the conditions. It's none of the fingers if you want to have the allele frequencies stay the same. None of the fingers. And then allele frequencies will stay the same. You will not have evolution. Since all of these things happen, allele frequencies change and evolution will occur. You got that? Okay, so the small finger was, a little finger is that you would have genetic drift. And the two forms we looked at were, tell me one more time, bottleneck and boundaries. Okay, and then the ring finger was for? Yeah, non-random mating. So you're picking somebody purposely to mate with. And the middle finger was? Mutations, which will change the DNA. Pointer finger is? Gene flow. So some genes coming in and some genes leaving. Okay, and the thumb was for? Natural selection. Good. Are you good on that? Okay. So, hey, you need me? No. Okay. So, since you can't meet those conditions, then the gene flow does happen. Do you want to go over those equations? Do you want me to review those with you?
Good.
You're good? Okay.
Should we know them?
And then the next part was focusing on natural selection and like why giraffes' necks would get longer, and that would be because you have a lot of babies. There's going to be what between the babies? Competition. There's going to be variations amongst the offspring, and those that are best adapted for the environment are going to survive and reproduce, yes? And you have lots of time. Then we talked about how the environment changes really rapidly sometimes, and what can happen as a result of that. It could increase some numbers and decrease other numbers, causing extinction, or you could have emergence of a new species. And we talked about what are the types of speciation, and there were two types of speciation. Remember the two types? One starts with an A and one starts with an S.
Allopatric.
Boom. Allopatric speciation. What was the other one? Sympatric. Good. So allopatric is there is some geographical barrier in a population, okay? There's some geographical barrier, and so as a result, they have their own mutations, and they're not mating with each other because of that barrier. They're having their own mutations, they're having their own selection pressures, and they have different initial gene frequencies. So as a result of that, they go their own way. So even if you remove the barrier, they've become so different they don't want to mate anymore. So that is one way, and that is the most common form of speciation, is allopatric speciation. All starts with a barrier. Sympatric speciation. Oh wait, here's a salamanders for allopatric. Sympatric speciation, an example on Lord Howe Island off the coast of Australia. This is where there was just new species right in the middle of a group of trees, and that probably was due to what? Chromosomes. Exactly. Due to chromosomes, some sort of chromosomal error, and so they have these new genes that are different, and they can't mate back with the parent offspring. Do you remember this picture? So what is this an example of? Why are the trees different on allopatric, and this would be sympatric? Perfect. And then we looked at another word called adaptive radiation, and the example there was Darwin's finches or the honey creepers on Hawaii, and it's like trying to fill every niche out there, and they exploit different niches, and they try to do resource, do you remember what? Partitioning in order to exploit those. And you guys, that was it. Yay, yes, go ahead.
For chapter 24, not 27,
Mhm.
can you give us like a few practice problems?
Yeah. Yeah, it'll be class lab. You missed the clickers?
Yeah.
Me too, because you could see the questions.
It's 9 to 6, 7 to 6, 7. I don't know the rest.
Miss Litton,
Yes, baby.
what's the class ID?
It is underneath that poster right. It'll come up. One more second, it'll come up.
Quinn, did you like those? Live?
Here's the activity code.
Miss Litton, on the final, are there
I reviewed that. I mean, there could be some. I'm not saying there's a lot, but it's on the review. Watch the Unit 5, Unit 6 review. Okay, no problem. There's a bunch of people in here from last. Okay, you ready? Do you want me to remove? I'm going to remove you because there's too many in there. Go again. Be whatever you want. Be yourself and be happy with it.
Lily. That's not the case.
GQ.
You better get
I remember that. I completely forgot about that until right now. Gee, you don't like GQ? It's a little magazine.
It's a men's modeling magazine.
Oh.
And he had his picture.
Oh, is it
was like,
Gentleman's Quarterly?
yes.
Oh, okay, got it. Yeah.
It's only one, but I'll pull up some more. There's some more right there. I can see them. Answer this one. Let's go. Is it hard?
No.
See, this annoys me. I can't even see what it is. I mean,
really-
I wrote it, but I don't remember. 5, 4,
We get it wrong.
3,
3, 2, 1.
bye-bye.
Okay, now,
Really got it wrong.
first of all, have I ever taught you the word DNA gyrase? No. DNA Elvis? No. So why would you pick that? You won. And then 3 of you said DNA ligase and DNA polymerase.
No.
Stop and listen. Yes, those are enzymes we know about. But no, that will not help you solve this problem, which is introducing DNA into another organism. DNA ligase, yes, it's about sealing, but DNA polymerase, it makes more copies of DNA. So we need something, if we're going to take a gene from one place to another, we need to cut and we need to paste. And that's why you would pick restriction enzyme and DNA ligase. Okay, let's do some more.
We have to reorganize them though because it's all messed up, so it looks bad. Yeah.
And then I have some more. This is how Five,
Hey, yeah.
4, 3,
Hurry.
2, 1.
Oh. Money.
Oh,
Yeah, good job, guys. Here's another one. Can I start it? I can. I need a nap.
we can't answer this.
Uh,
It says who committed the crime.
It's
Oh.
subset.
Possibly.
Oh,
C3.
unable to tell. Oh,
There's guys, stop. You're making judgments you shouldn't make.
who are we comparing it to?
B.
B.
Whatever that one is.
That's not an option.
No.
Which one is A and which one is?
It's B.
We have the option of 1, 2, and 3.
Suspect.
So like put them in order.
There's definitely
It's 2.
Go 3.
Money.
because there's 12 reproduces.
I don't think you.
Oh, that's the crime scene evidence. That makes sense.
Yeah,
I mean, we
that's
got it
not
wrong.
the perpetrator of the crime.
Anyone want more liquor?
Benny's really hungry.
So we know, yeah.
Butterfingers.
That's it. Okay, Smarty McSmarty Pants,
Wait,
be
what were
ready.
the equations you're gonna show us?
Oh, the equation, if you remember.
S plus Q equals 1. S.
Oh, P plus. Okay.
This is your gene pool, okay?
Gene.
Can you write that up there?
Gene pool.
Right, gene
Can, yeah,
pool.
can we write gene pool up there?
Yeah, gene pool. Yeah.
Thanks.
There we go.
That's gonna be a good
What
picture.
comes out of our gene pool?
P squared.
P squared, which would be homo dom,
Oh, these are pretty good.
Nice.
which is hetero dom.
Yes. Good stuff. Boom. Doesn't it equal 1?
Whoa.
Yes, it does.
There we go. There you go.
Alright.
Straight up.
They're gonna be good though.
Good job, guys. I'm proud of you, and I love you very much, Lee.
How many questions is it? Like 70?
81.
Yes.
Have a piece of toast.
And a couple like free.
1.
And then
Free
just
one.
do 40 or 20 on the biomimicry.